Welcome back, gamers, to episode 3078 of my Minecraft Let's Play. I thought we could talk about DNA today, because DNA are the building blocks of life. But first, lab safety. This is a cell. You have a lot of these, and there are some important things in here. Plasma membrane, cell nucleus, Golgi apparatus, your ribosomes, endoplasmic reticulum, and cytoplasm. And also your mitochondria, but that's not pictured here. But we're not going to be focusing on all that today. We're going to be focusing on your cell nucleus, because that's where your nuclear deoxyribonucleic acid, or nDNA, is stored, and that's the majority of your DNA. Some might be in the mitochondria, where it is called mDNA. Inside the cell nucleus is a chromosome. You have 46 of these that pair into 23 pairs. They sort of look like two elytras facing each other. Let's take a closer look at the chromosome. On the bottom, you have Q arms, which are the long arms. And at the top, you have the P arms. And these are connected with a centromere. To protect the DNA stored inside this chromosome, there are telomeres, like caps. Your DNA is being protected because chromosomes are actually just wound up strings of DNA. So what are these strings made of? Well, basically, this thing called base pairs, where it's two elements that bond together. The base pairs bond to the sugar-phosphate backbone, represented by the blue blocks. Here is adenine and thymine, represented by amethyst and terracotta. They bond together. Then there's guanine and cytosine, which are represented by green and cyan concrete. To understand how these bond together and why they do, we actually have to go into the chemical structure of these elements. Looking at adenine and thymine currently, you can see that there are hydrogen in the chemical compounds. These hydrogen form hydrogen bonds, as pictured here. There are only two bonds between adenine and thymine, while guanine and cytosine have three hydrogen bonds, making them the stronger bonded base pair compared to adenine and thymine. These base pairs, then bonded to the sugar-phosphate backbone earlier mentioned, will then twirl up into what is called a double helix structure. The reason it winds up into this double helix structure is, in essence, because of storage and ease of transportation. It's important to note that while the cell is not preparing to replicate, the DNA is actually unwound. A way to think about it is if you were to send a zip file to someone and they had to unzip it or decompress it. Fun fact, it's actually debated who discovered the double helix structure. But back to the important part. So why should we care? Well, a few reasons. Most obvious of all is to understand chromosomal disorders, but also the future of vaccines is really important in relation to DNA structure. And also for the future of gene editing, in which DNA is changed through a myriad of ways. And the important part of it is that it can help treat or cure genetic diseases or those acquired. The most important reason is actually because if we know where and where we come from, we know where we can go in the future, and it opens up so many possibilities.